With over 45 years of cell culture experience, ATCC never forgets that the most valuable asset to your research is healthy cells. That's why the leaders in cell line authentication have developed the new Universal Mycoplasma Detection Kit, a unique tool that includes essentials for fast and reliable sample preparation and PCR, together with detailed instructions, lysis buffer, universal PCR mix, universal primers, sample lysis tubes, and a positive control. Scientists at ATCC know that mycoplasma remain a significant source of contamination in cell culture, costing researchers time, money, and resources that could have been saved by routine screening. With an easy-to-use protocol and providing results in less than 3 hours, the ATCC Universal Mycoplasma Detection Kit affords frequent testing of cell cultures for solid peace of mind. Mycoplasma are small and rarely produce turbidity in cell culture. The superior sensitivity of the kit detects less than 10 genome copies of M. arginini in a standard assay. And the combination of a unique universal enzyme and an optimized touchdown PCR program produces finer specificity, eliminating non-specific amplification of cellular DNA. Results are highly selective, recognizing over 60 species of mycoplasma, acoloplasma, spiroplasma, and ureaplasma, including the 8 species most likely to contaminate cell cultures. The ATCC Universal Mycoplasma Detection Kit is a fast, easy, and affordable way to safeguard the most valuable asset to your research. To learn more, visit us online at www.atcc.org/mdk. And never forget to love your cells with the frequent use of the ATCC Universal Mycoplasma Detection Kit.